There we go. Okay, so the soma is essentially determining the all or none response, all or none principle. So when these chemicals enter into the dendrites to start, the neuron's at its resting potential, which basically means it's not doing anything. But if the right kind of chemicals enter the dendrite, then the soma is going to react with those chemicals and make a decision to turn that all or none response and change from the resting potential to the action potential. The action potential now meaning the neuron's doing something. So that's the soma's decision. It's taken in these chemicals, and it's decided that it's going to do something. When the soma decides to do something, that's when it changes from a chemical messenger to an electrical messenger. Meg.
Is this the diagram that we're supposed to like?
No, not exactly like this.
Okay.
I don't have you label all of these things.
Okay.
But just in relationship to questions, I might give you just a picture of the neuron and ask you to label those things, or I might ask you to explain something that's happening in one of these pictures. Right? In relationship to this section, this would likely be labeling or short answer. Okay. So we have all these chemical things going in at the dendrite. So I'll just put an arrow. So there's chemical. And then let's say that the right chemicals came in, and so the soma decided to turn the neuron from its resting potential to its action potential, which means then that an electrical message gets passed down this axon. Electric. It's electric. Right? So this message is going down the axon, and the axon, so that's kind of like your step 2. So step 1 is going on all at the top. Step 2 is happening in the axon. There's this thing called the myelin sheath that's covering the axon. The myelin sheath has 2 functions. One, it speeds up the process. So for example, if we think about a regular wire in your house, when you're trying to connect cords, like maybe your power cord from your TV to the wall, you don't have just like raw copper wire that you're playing with. That would be extremely dangerous. It's covered with that insulation. Well, the myelin sheath is doing the same thing. And it does 2 things. One, it helps speed up this transmission. So it makes that electrical message move more quickly down the axon. But it also is protecting the axon, just like plastic around the outside of copper wiring would protect it as well. The myelin sheath is made up of these things called glial cells. Glial cells. That's what the myelin sheath is made of. And the myelin sheath, these glial cells, it's literally meaning glue. And so it's holding together the axon, it's holding together the process. So particularly issues and disorders like MS, multiple sclerosis, what's actually happening in them is the myelin sheath is breaking down. And so information is not transmitting properly between people's muscles. And so the messages aren't actually communicating properly with different parts of the body. And that's just relating to just one of many biological disorders that are out there.
Should we write that down?
You should write down glial cells.
Okay.
Yeah. That's one of the words on the list. I just don't, well, here, I can write it, I guess, here.
No, it's fine.
Okay. All right. So then, we get that electrical message. Yeah, thank you. We get that electrical message to the bottom. And these are what are called the terminal boutons. It's not buttons, it's boutons, it's French. And the terminal boutons are taking this electrical message. And this over here is a screenshot, or a close-up, I guess, of the terminal boutons, okay? So we have this electrical message that came down. And that electrical message pushes these things called vesicles. And these are the vesicles, to the walls of the terminal boutons. Now, inside those vesicles are these things called neurotransmitters. And the neurotransmitters are the chemicals. So remember that with the dendrite, we started with a chemical, and then we changed it to an electrical impulse, because electric can move a lot faster than a chemical can. So if we're actually looking at being effective with our bodies, the electrical message moves much more quickly. So this electric message basically pushed these vesicles out and pushed them to the side. And that released the neurotransmitters, which are the chemicals. So let's see, how about, oh, green, I haven't used green. We'll do that. Yes, thank you. Okay, so in here, this is kind of like your fourth step that we're into. Okay, and this is where we have these neurotransmitters, which are the chemicals. So those chemicals moved into what's called the synaptic cleft. And the synaptic cleft is this space between the neurons. So if I go back to my black to show you, this piece that we're connecting to is a dendrite. We're back to step one. So what's happening is we're going through the whole process. We're starting all the way at the top, we're moving all the way down, and then we're just reconnecting to a new one and redoing the process again and again. So it's like a constant relay system of information that we're getting.
Like the ones that weren't used?
That's, we're getting right there.
It's like recycling.
Yes, yes, okay. So we're going to focus a little, we're going to talk a little bit more about what's going on in the synaptic cleft, okay? But again, I want you to make sure that you're recognizing we're starting with chemicals. Those chemicals are triggering the all or none response to either say, hey, not enough chemicals or not the right kind of chemicals, so we're not going to do anything, or hey, yeah, we do want to continue this message, and so we're going to move into the action potential. That action potential is an electrical message that moves down the axon, which is covered by the myelin sheath, which is made of glial cells, and then it ends up in the terminal boutons, which are holding these vesicles with little neurotransmitters in them. And the neurotransmitters are those chemicals. So that electrical message moves into the terminal boutons and pushes those vesicles to the edge of the terminal boutons and forces the neurotransmitters into the synaptic cleft. Now, once these neurotransmitters are in the synaptic cleft, there's three things that can happen to them. And one of which, Caitlin, is what you said, where they get used back up. Okay.
Are you going to have this available in like wording?
I'm going to take a picture of this in words. That's in your book.
It is? Okay.
But like Jessica was saying, it's, you know.
Kind of wordy.
Yeah. So you have your book, you have this,
Okay.
and
If you want, I'm going to put this on my Google Doc,
Are you taking a video?
so you can
we're going to go
actually send me
ahead
an
through
email.
the three things that can happen to the neurotransmitters once they're in the synaptic cleft. So the one thing that they can do is they can connect to another dendrite, and they're like keys. So each neurotransmitter has its own shape, and the dendrites on the other side have little spaces. And those spaces only accept certain neurons. So one thing is that the neurotransmitter can essentially fit into a receptor site on another dendrite. And if it does that, if the neurotransmitter makes it over to a new dendrite, and it fits in, and it goes, yay, it matched. Well, then now it's a new chemical restarting the process over. That's the beginning of it. Not all neurotransmitters, though, are going to make it to the next dendrite. So the next thing that can happen, yes, it can be rejected. And when it's rejected, we can either have the neurotransmitter break down, or it can be recycled. Okay, so another option that can happen is that they don't get chosen, and they get broken down by enzymes. Now there are lots of very specific types of enzymes. If you're interested in looking it up, you can definitely find out about it. That's beyond the scope of what we want to cover. But if they're broken down by enzymes, they're not being used anymore. The third thing that can happen, though, is that the neurotransmitters can go through a process called reuptake. And that's like the recycling process. So reuptake is when those neurotransmitters go back into the vesicles. And so they get repackaged back into the original vesicle so that way they can be reused again.
And they're the ones that matched and fit and stuff, or the ones that weren't used?
They're the ones not used but also not killed. So the ones that do get used are the ones that are fitting. If they don't get used, they either are killed, right, or they're repackaged. That's fine.
So they're either repackaged or they linger, or is it a combination of the two?
One of these three things will always happen.
What I mean for
yeah.
just number 3.
Yeah.
Because it mentions like how they can either linger in the synapse or be put back into a
It goes
vesicle.
back in vesicle, and then it just.
Yeah, so they hang out in here until one of these three things happen.
Oh, okay.
And
All of them.
right. So one of those three things is always going to happen to them, and then depending on perhaps other drugs that are in the system or what they're trying to connect to, they can linger a longer or shorter period of time. But one of those three things is always going to happen, and exactly one of those three things. So I'm going to go ahead and post this picture for you as well as the PowerPoints. We've now hit all of those terms on that fourth snapshot question. Digest everything, okay? Real quick. Okay, I'll of course we'll be back on Tuesday. Again, I strongly encourage you to do the